Pronuclear microinjection. In the last slide, we discussed the outline that how the micelles they can be transfected कि किस तरह से एग की ट्रांसफॉर्मेशन की जा सकती है या जो एम्ब्रियोनिक सेल्स हैं उनको किस तरह से ट्रांसफॉर्म करवाया जा सकता है और कौन सी टेक्निक्स इसमें यूज़ की जा सकती हैं. So one of the methods is that the pronuclei in the fertilized egg they may be the target. That pronuclei they can be transformed with exogenous DNA by using a microinjection technique. First, we should know that what is a pronuclei. Pronuclei they are the egg or the sperm nucleus के जो एग के अंदर या स्पर्म के अंदर जो न्यूक्लियस होता है उसको हम प्रोन्यूक्लियस कह सकते हैं. And microinjection technique has been introduced to inject foreign DNA into one of the nuclei, that is either female or the male nuclei. And microinjection technique has been first time used to create transgenic mice by introducing the foreign DNA. And the first attempt that was made, it is by using the DNA from a virus which is called as SV40. So the DNA from SV40 was injected into the blastocyst of the blastocyst. Blastocyst, it is a structure that is formed after fertilization of the egg at the later stages. And it contains a cavity which is called as a blastocele. तो SV40 का DNA लिया गया और इसको इंजेक्ट किया गया ब्लास्टोसील के अंदर. तो फिर जो एम्ब्रियोनिक सेल्स थे या ब्लास्टोसील का जो स्ट्रक्चर है पूरा उसकी इम्प्लांटेशन करवाई गई जो फोस्टर मदर है उसके यूट्रस में. फोस्टर मदर का कॉन्सेप्ट हम पहले भी डिस्कस कर चुके हैं कि ये एक्चुअल मदर नहीं बल्कि ये मदर का जो किरदार है वो अदा कर रही होती है. तो इसमें हम एक मदर से या डोनर मदर से ब्लास्टोसिस्ट लेते हैं, उसको ट्रांसफेक्ट करवाते हैं और ट्रांसफेक्शन के बाद हम ब्लास्टोसिस्ट को हम फोस्टर मदर में इम्प्लांट करवा देते हैं. एंड आफ्टर इम्प्लांटेशन some of the cells of the blastocyst they may be transformed while other remains unaffected. So sometime what happens that if the foreign or exogenous DNA it has been integrated into the host genome, it can result the formation of the transgenic mice lines after or after cell division or even in the next generations. It is not only the blastocyst that can be transformed by using the SV40 virus. Rather, egg cells they can also be or fertilized egg cells can be transfected by using the microinjection technique. And one of the microinjection technique that we are going to discuss it is the pronucleus microinjection. Now, in this technique, after fertilization, male and female nuclei they remain separated for some time. That is, just after fertilization, here because of the larger size of the male nucleus, it is selected for the microinjection of the foreign DNA. And then after the injection, it is implanted into the foster mother. Now, what techniques it is being used for the microinjection of the pronucleus? This is an egg cell that contains two nuclei. Suppose it is a female pronuclei and it may be a male pronuclei. So male pronuclei, because of its larger size, it can be selected for transfection. And here the expert eyes are required to handle the egg cell and the needle. So as I told you that after implantation, now the cell lines either they can be transfected or some cells they remain unaffected. But overall we can say that the transfection efficiency it may be 5 to 40%. So this is one of the method by which the fertilized egg cells they can be transfected.